Thank you very much, Sonia, for introducing me, and thank you for having the possibility to present in the framework of ZMT Reef eTalk my work. So the project that I'm talking about today was actually conducted during my time at the Australian Institute of Marine Science in cooperation with Katharina Fabricius. So as most of you will know, coastal areas are under increasing pressure from human population growth, intensified land use, and urban and industrial development. As well, increasing temperature due to rising greenhouse gas concentration lead to increasing rainfall variability in many tropical regions. And as a consequence, more suspended sediments, nutrients, and pollutants enter coastal waters. And this you can actually see in these pictures down below. So here you can see big flood plumes from rivers, and here pollutants and sediments entering the coast from smaller river systems. And this actually leads to a decline in coastal marine water quality and clarity. In the Inshore Great Barrier Reef, land runoff is the largest source of suspended solids and nutrients. And along the coast of Queensland, more than 30 major river systems and numerous smaller river systems suspend their sediments and nutrients into the coast. And here you can see on the satellite image the flood plume of the Biochemical River of the Queensland coast in February 2008. And I think this is quite impressive how much sediments actually enter the coast, which not only leads to an increase of particulate nutrients and sediments into these environments, but also it strongly reduces the light availability. And on the left-hand side, you can see coastal environments impacted by these flood blooms of the Biochemical River, and shaded in dark blue are the ones which are mostly affected. So corals living in these environments need to have species-specific abilities for adaptation and acclimation, which enable them to survive and thrive in these environments. And this slide illustrates that adaptation actually happens on the longer term, because it includes changes of the genotype or the genome, while acclimation includes changes of the phenotype at a given genotype in response to changing environmental conditions. And in the literature, it is strongly debated whether marine organisms will be able to acclimatize to the rate of ecosystem change that is happening at the moment and that is predicted for the future. Nevertheless, a high phenotypic plasticity, meaning the potential to change the phenotype, which can include physiological traits, morphological traits, or behavioral traits, is a very important strategy for marine organism to be able to withstand changes in an environmental condition throughout their lifetime. So acclimation can take actually place throughout the lifetime of an organism. So to date, it remains very difficult to estimate the actual effect of increasing land runoff on coral reefs. And the literature reports very contradictory effects, ranging from beneficial effects due to the fact that higher nutrient contents can lead through heterotrophic feeding of the corals to an improved nutritional status. Experimental studies have also shown that well-fed corals can increase or showed higher resistance and resilience towards increasing temperature in comparison to experimentally starved corals. They also showed a higher photosynthetic efficiency and higher cell protective capacities like antioxidant enzymes. On the other hand, we have a lot of reported negative effects ranging from a loss of coral diversity and cover to altered species composition, shallower depth distribution limits due to the decrease in light availability for the corals. And these effects are mainly due to a reduced recruitment success, decreased calcification, greater bleaching susceptibility, higher oxidative stress, and lower photosynthetic efficiency. And according to the reported negative effects, the coral cover of reefs around the world is decreasing, and that has also been shown for the reefs in the Great Barrier Reef, and especially in the inshore region. Inshore reefs, the coral cover decreases already since more than 3 decades. And the Reef Rescue Marine Monitoring Program, which is run by colleagues from Ames, they found that in the Burdekin region, so along the coast of North Queensland, especially since 2009, the coral cover showed a steady decline. And Thompson, who is one of the team members of the Reef Rescue Marine Monitoring Program, he did a study where he investigated different genus of corals, and he looked at the juvenile abundancy and the coral cover within 10 years. And these 10 years included a 5-year period below median river discharge from 2002 to 2006, followed by 6 years of above median river discharge from 2007 to 2012. And he found actually that especially Acropora was very sensitive and showed steady decline over the whole time of investigation. And the decline in both in juvenile abundancy and coral cover was more pronounced at 5-meter depths compared to 2-meter depths. So in order to better understand why coral cover declines and what are actually the physiological consequences of the exposure of corals to higher turbidity, higher nutrient content, and diminished light availability, I investigated Acropora tenuis as a model species, which you can find in inshore reefs, but also in offshore reefs. And I investigated them here at Jeffrey Bay, which is a high turbid reef site at Magnetic Island, for those of you who are familiar with the region around Ames. So directly in front of Townsville, only I think around 8 km away from the shore. I also investigated physiological traits of corals here at Pelorus Island, which is a low turbid reef site, so you can see that it's still exposed to the flood plumes of the Burdekin River. And then I also looked at the performance of corals at Davis and Rip Reef, which are 2 corals more offshore, like 50 to 65 km offshore, and distant from the flood plumes. And here I would like to give you an idea about the environmental parameters at these 4 sampling sites in 5-meter depths. Here Jeffrey Bay and Pelorus as the inshore reefs, and they actually show, you can nicely see that that light availability is very low here in these inshore reefs and increase when going towards the midshelf reef. So they have much more light available throughout the year. And you can also nicely see that the content of suspended solids, but also particulate nutrients like phosphorus and nitrate, which is not shown here in this graph, but it was a similar picture, they have highest contents here in the inshore reefs, as well as the chlorophyll A content was also higher when going towards the land. And what is also obvious is that you have a strong seasonal effect. So during the wet season, you have really very, very low light available here at Jeffrey Bay. So it's much lower than in the dry season. Same for Pelorus Island. Here you have also the strong seasonal effect of light availability and contents of sediments, chlorophyll and nutrients in the water, whereas the effect is less strong here in the midshelf reefs. So we collected coral branches at these 4 reef sites between 2 and 5 meter depths. We snap froze some of the branches to look at biochemical parameters as a measure for cell protection. So we looked at the total antioxidant capacity. We measured apoptosis rates, which gives you an idea of cell damage and the need to renew cells which have been damaged, and also the chlorophyll content, which are photosynthetic active pigments of the corals. I also incubated live coral branches in these chambers that you can see here. So this is like a closed system with a defined water volume, and here there's usually a stir bar in the bottom which provides the coral during the incubation with water flow. And I incubated these corals both in the light and in the dark to measure net photosynthesis, dark respiration, as well as light and dark calcification rates. And the inshore corals were exposed to light levels, which you can usually find in the field. So they were exposed to 200 and 350 micro-Einstein, whereas corals which live in more clearer water in the midshelf reefs, they were exposed to 350 and 550 micro-Einstein during the incubations. And here you can already see the results. And so this is net photosynthesis and light calcification. So the light dependent physiological parameters measured in live coral branches. And you can see that the productivity, including net photosynthesis and light calcification, was actually highest at Jeffrey Bay during the wet season, which was in the first place surprising. But then when we looked at pigment content, it became clear that these corals are actually very well acclimatized to the more turbid conditions. And this is why they actually perform really well. On the other hand, when reducing the light availability, you can see that during the wet season and the dry season, and at both reefs, Pelorus shown in lighter gray and Jeffrey shown in darker gray, that productivity is reduced by sometimes by more than 50%. So if you imagine a flat plume during the wet season, corals will be actually strongly impacted. And this can lead to a reduced skeletal density, which was shown by Melissa Rocca in her study here, published in 2017 in the Marine Pollution Bulletin. I worked with her together at the different reef sites. And so here in these graphs, you can see the performance of corals at Jeffrey Bay, the most turbid reef site, Pandora Reef as a middle turbid reef site, and Pelorus Island as a low turbid reef site, here represented in the different shades of brown. And you can see that the performance of the corals at the more turbid reef site, when looking at growth or at the lipid content, is actually really good. So they are actually well acclimatized to these conditions. But here, when looking at the skeletal density, you can see that it's really reduced at the more turbid reef site compared to to the low turbid reef site at Pelorus Island. And this is due to the fact that the skeletal framework usually grows, like extends during the night, during dark calcification, where crystals are randomly growing. And then this framework usually gets filled in during the light. So as here, the light calcification was impacted, we can assume that the dark calcification was still pretty good because the growth was not impacted, but the framework gets more brittle, and this will actually increase their susceptibility to physical damage, for example, during cyclones. So here they are really, they show a really impact of the lower light availability. And here you can see that I added the midshelf reefs to the picture. So just simply due to the fact that they have more light available, they can be much more productive at the midshelf reefs. While you can see that when exposing them to the same light level, they show the same level of productivity. But during the wet season, where light is usually strongly diminished and 200 is like an average value, we can assume that both net photosynthesis, so energy gain, as well as light calcification will decrease. As I mentioned before, and what we expected that, and we could also see that, was that the light independent parameters here, dark respiration and dark calcification, we could actually see no differences between the reefs. So dark calcification was similarly strong during the wet season in all at all 4 reef sites in the corals. Also, other biochemical parameters that I measured were similarly high in the wet season and in the dry season at all 4 reef sites. So the cell protective capacities were similarly high at all 4 reef sites, as well as the rate of apoptosis, so the need to replace damaged cells. The only thing that we could see that was the midshelf reefs. They slightly upregulate their total antioxidant capacity, and this is probably due to the fact that you have higher levels of irradiance during the dry season in the midshelf reefs, and this actually can increase due to higher photosynthetic activity, the level of ROS production, reactive oxygen species, and they protect their cells by upregulating cell protective capacities. And as you can see here down below, the level of cell damage is not increasing due to this higher protection by antioxidants. And here are already my conclusions. So Acropora tenuis has the potential to acclimatize to inshore conditions, and especially the light independent parameters that we investigated did not differ between all 4 reefs. Nevertheless, both net photosynthesis and light calcification will be affected in corals living at the more turbid reef site, especially during flood events. And this reduced light availability lowers light calcification, which decreases the skeletal density and which makes them more susceptible to physical damage. What we have to keep in mind is that during the time, like when we studied the corals in 2014, we had relatively mild conditions. So it was a dry year. We didn't have like a strong flood event. And also the year before in 2013 was relatively mild. So it would be interesting to really measure the physiological performance of corals during a flood events, or in years where you have like where you had several strong wet seasons in a row. And this is already the end of my presentation. I think I was quite quick. I would like to thank you for your attention. And I would like to thank all my colleagues at Ames who supported me, and it was a lot of fun to work with all of them. So especially the MMP team, parts of my working group, Sam and Sven, the crew of the Cape Ferguson, the Ames workshop, and students who worked with me in this project. And of course, I would like to thank the funding organizations. Yeah, and now I'm happy to answer any questions.